Hi, thanks for joining me at Student Nurses Success, where we're going to cover a video on genetics. We're going to cover chromosome disorders, congenital disorders, and Mendelian Single Gene Disorders. Remember, you can find a link to the maps in the comments section. Let's go ahead and get started. I'd like to start with our blank map. We'll first begin with just some terms. Genetics is really the study of inherited traits. Chromosomes carry the genetic material, and on the chromosomes are genes. These genes are located on specific locations or sites on the chromosomes. So they would be located like here's a gene, here's a gene, here's one, and here's one. Now, genes are the instructions for the traits. Alleles are in two different forms, and they are different instructions for the same gene. With alleles, you inherit one allele from each parent. If you're homozygous, then both alleles are the same, and if you're heterozygous, then the alleles are different. A mutation is a permanent change in the DNA sequence of a gene, and mutations can occur spontaneously or they can be inherited. The karyotype is your actual genetic code or the complete picture of the genetic code. So here I have a picture. This would be a karyotype down here, the complete code. And a phenotype is how traits appear on the physical body or the physical expression of the code. For example, whether or not you have green eyes or blue eyes. When we're talking about multifactorial with regard to genetics, we know that environment plus genetics plays a role. So it's not just genetics and just not just environment, but the combination of the two play a role in a genetic disorder. That's said to be multifactorial. Polygenic means there's more than one gene that determines the presence of the disorder. Let's go up to chromosome disorders. Chromosome errors usually occur during meiosis or in early cell division, and it actually usually occurs most commonly during the crossing over. During the crossing over of meiosis, we can have breakage or errors where maybe the genes flip, or one gets missing, or one gets duplicated too much. Trisomy 21 or Down syndrome is a disorder where there's an extra copy of the 21st chromosome. Now, there's other disorders like trisomy 18 or trisomy 11, where there would be an extra copy of those chromosomes. However, those are more rare, and a lot of times the fetus does not make it to term or dies shortly after birth. With trisomy 21, it's more common, and we see more cases of it. We do know that trisomy 21 is associated with advanced maternal age of the mother, and that we know as eggs age, they're more likely to have this error. Manifestations of trisomy 21 include the presence of epicanthal folds, a simian crease. If you look at your hand, you have three creases in your palm. A simian crease would be one. Intellectual deficit, which can range from mild intellectual deficit to severe. Characteristic facies or a characteristic look to the face, and short stature. We know that individuals with trisomy 21 are high risk for having congenital cardiac defects when they're born. They get increased respiratory infections and have a degree of immunocompromised status. They are higher risk for leukemia and early onset dementia. Now, Klinefelter syndrome is when there is an extra X, but there is the presence of a Y, and that determines male sex. So you might see XXY or XXXY. With Klinefelters, we see a delay in puberty, decreased testosterone levels, feminine features, including gynecomastia, and a tall, thin stature. Treatment for Klinefelters is testosterone replacement. Turner syndrome is similar, except for you do have the extra X's, but there is no Y, so that is a female sex. So XXX or XXXX. Manifestations include short stature, webbed neck, decreased estrogen, ovarian failure, failure, increased cardiovascular risk, probably from the decrease in estrogen we know, and osteoporosis as well. Now, congenital disorders, we know that inherited genetic disorders that appear later in life are not necessarily considered congenital. When we're talking about congenital, they are things you are born with. A congenital disorder could be several mutations or it can include a condition. A congenital malformation is considered a structural defect. We know congenital disorders can be genetic, they can be inherited, or they can be from environmental exposures during pregnancy that cause the congenital defect. Teratogens are factors that cause malformation or disorders. Um, they cause congenital malformation or congenital disorders. A teratogen could be a chemical exposure, exposure to some type of pollutant, maybe exposure to medication or drugs during pregnancy, contracting infection during pregnancy or an illness, or exposure to radiation. So, any exposure while pregnant can be considered a teratogen and can harm the developing fetus. We know that weeks 3 through 12 are our highest risk of vulnerability for the fetus. Exposure less than 3 weeks may damage the developing organ so much that the baby is not viable, or they may not really, because nothing is really formed that well in less than 3 weeks, it may not really have any impact. We know that after 12 weeks, most organs are fairly formed, so there's less impact. Porch are a group of infections that, when contracted by the mother during pregnancy, um, actually cause high risk of congenital disorders in the baby. These include toxoplasmosis, which is from cat litter boxes, HIV, Zika, which can cause microencephaly, which is a small head in the baby, syphilis, rubella, CMV, and herpes. Let's move on to Mendelian single gene disorders. Mendelian single gene disorders follow a very predictable pattern of inheritance. There's a mutation on a single gene, and we know family genograms are really helpful in identifying this. There are a few rules with Mendelian single gene disorders. One is that single genes randomly and independently are transmitted. Each pregnancy is its own new event, so it's not related to a prior. So if you have a 1 in 3 chance or 1 in 4 chance of having a baby with this disorder, that each pregnancy is a 1 in 4 chance. 50% chance of receiving one gene from each parent. Now let's talk about autosomal dominant disorders. In autosomal dominant disorders, it only takes one gene to express the disorder. We know that males and females are equally affected, so it doesn't matter. It isn't attached to an X or a Y chromosome. The affected individual must have an affected parent. And if you do not have the disease, you do not transmit it to your offspring. Common examples include Marfan syndrome, which is connective tissue disorder, Huntington's disease, which is a neurodegenerative disorder, or polycystic kidney disease, which results in a kidney failure. Let's do a Punnett square for Huntington's. Let's say dad has Huntington's, and let's say mom does not. When we do the Punnett square, what we will see is a 50% chance that any baby will have Huntington's because it only takes one gene to express it, and a 50% chance they will not. So again, mom doesn't pass that on, but dad does in this case. Now let's talk about autosomal recessive disorders. With autosomal recessive, we know that you need two genes or you need to have both in order to express it. With autosomal recessive, males and females are equally affected. It does not have anything to do with the X or the Y. A person can be a carrier, meaning they just have one gene for it, and they may not even know that they have it because they don't express the gene if they are a carrier. So one gene makes them a carrier, but they do not express it. They just pass it on. We know that consanguinity increases chances. And when we're talking about that, that we want genetic diversity, and that's why it's not a good idea to keep your gene pool small. So in situations, that's why we don't marry cousins or brothers or sisters. Common examples include sickle cell anemia, where hemoglobin quality is poor, cystic fibrosis, which is exocrine gland dysfunction that really causes excessive mucus in the lung and lung infections, and phenylketonuria, which is a metabolic disease where you're unable to metabolize phenylalanine and it builds up in the bloodstream, poisoning the brain. Let's look at a Punnett square for autosomal recessive. Let's say cystic fibrosis. And in this case, let's say mom is a carrier for cystic fibrosis, and let's say dad is a carrier for cystic fibrosis. So they are both carriers. Neither one of them know that they have this gene. And so it does become a bit of a surprise. It is a 1 in 4 chance or 25% chance that they could have a baby with cystic fibrosis, the disease. A 25% chance they would not have the a gene at all, and a 50% chance that this couple would produce a carrier. Our final one I want to talk about is X-linked recessive. So it's recessive, and so there is no dominance. You would need to actually, if you have a secondary gene that would counteract it, you would not express the disorder. But in X-linked recessive, it is caused by a gene on the X chromosome. We're not going to talk about Y-linked recessive disorders. They're much more rare. Now, with X-linked, females tend to carry because they have a second X, so they may not express the disease unless they have both of their X's affected by it. But males, because they only have one X, if they have it, they will express the disease. They don't have a balance for that other X. So an effective male will pass on to the daughter because the male passes the X to the daughter, making her a carrier, and fathers do not pass it to the son because they pass their Y to their son. It's mothers that pass it to the son because they pass their 1X there. Common examples include hemophilia, which is a bleeding disorder, color blindness, or Duchenne's muscular dystrophy, which is a neuromuscular disease. Let's run through an example here. Let's say we have a mom who is a carrier for disease X. And so let's say she's a carrier for disease, well, we'll call it disease D. And let's say dad does not have it. So in this particular case, it's kind of hard to fit these in here. Let me run these for you, and then we can explain them. Okay. So in this particular case, when we look at girls, we see that of the girls, there is a 50% chance this girl will be a carrier, and a 50% chance that the girl will not carry the gene at all. But she will not express it because there's only one there. For the boys, we see a 50% chance that the boy will have it because they do not have a balance for that. other X and that one X has the disease, and a 50% chance that they will not. There you go. There is our completed genetics map. Remember to subscribe and like to keep getting our videos and our updates.